Right, so hello everyone, and today we are going to talk about DNA's 1000 dresses. Now, of course, this sounds like a very elusive title, and indeed, my first title idea was how does the idea of different clothes for DNA help physicists make sense of DNA's biological functions? So you see, it's a bit less catchy, much longer, and what it said though is that we are going to talk about physics and DNA. And before delving into these details, what we are going to do is basically contextualize a bit and take a step back. So if we want to find out what is the popularity of a concept in a given culture, how would we do it? One way would be to propose to look at the frequency of usage of the corresponding word in the said culture. So we are in the UK, so we can look at something called the British National Corpus, which has a database of 100 million words and builds statistics of word usage based on this. We are interested in DNA, so we want to look at how frequently is it used. This is used on average 3,310 times. But then we need to put it in perspective, obviously, because DNA appears quite technical. So let's put in perspective with a common word. So we'll put it in perspective with a word that has been used 3,316 times, so slightly more. But the word in question is beer. And that's in Britain. So indeed it seems that DNA is very popular. Now, this popularity was illustrated and explained in 2003 by Steve Connor in an article listing 23 ways in which DNA actually changed the world. And here I want to highlight a few of them. So first, there is this idea that it provides a molecular explanation for trait inheritance, so from parent to child, which was lacking before. Second, it unlocks new developments in medicine, which is, of course, always welcome. Third, it may hold the secrets to the origin of life on Earth, which has to do with existential meaning. And also, it enables a fairer justice system. So these are the four that I would highlight from this. But that was in 2003. Now things have happened since then, and in particular there is the evolution of technologies that are based on DNA, where we build devices or self-assemble even devices out of DNA. One of them, these devices could be as well a memory storage device, which would be much greater in capacities than what we have at the moment. Right, now what is DNA? So DNA stands for deoxyribonucleic acid. What it means to a physicist is that it's very complicated biochemistry. And it's not that I don't want to go into it, but it's just that it would take too long for this particular talk. However, we can try to provide a simple, if not simplistic, representation of DNA. So it would be represented with two strips, say, like here in blue and red, and they are kind of attached together via these links here in dark. This blue here would be a DNA strand. What it tells us is that DNA can exist as a single strand of letters. And these letters here are the genetic alphabet, made of the genetic alphabet here, so A, T, C, and G. Now, if you will, you can interpret this blue strip as being an actual book itself. And then the red strip is somehow the negative of that book. So it's a copy of that book, but a carbon copy, kind of. And this is called the complementary DNA strand. Now, for things that have to do with the way nature works, these tend to be bound together, and for some reason as well that have to do with so-called steric interaction and various other kind of interaction between two successive pairs of letters, so let's say CG, CG, like from the right-hand side, they don't want to be exactly aligned. They prefer to be slightly mismatched like so. And so this gives them a slight twist, which basically gives them the shape of the familiar, let's say, double helix form that we are used to. Now, what can physics bring into this discussion? Well, if we take physics like any other creative activity, which consists in looking at the world through particular lenses and with their own set of filters and focus distances, then physics can look at everything, right? And in particular, I just want to give an example with sports, okay? You can look at the physics of sports. In this particular case, what people will look at is how can the rules of physics, as we understand them, give rise to the observed phenomena. And the three that I've proposed here, are in particular for sure the last two, are still very much debated, okay? And so the point here is that regardless of whether this is successful endeavor, usually this won't change the actual way the sports are practiced, okay? So it's really an additional perspective to it. So here I want to do the same thing but for DNA. There are some phenomena relating to DNA that we are interested in here. So the first one would be efficient compaction. So there are about 2 meters of DNA in each human cell, or at least in almost each one of them, which is a lot, right? This is about a micron in size, so millionths of a meter. Now, DNA acts as well as some form of Rosetta Stone of biology. Somehow, all molecules kind of go onto it and somehow can read some aspects of it. So it can be read by many different molecular machines. Finally, it has some form of context-dependent expression, so it can make some of its information accessible only when required. So then what we are asking is how can the rules of physics, as we understand them, explain this versatile combination of DNA's functional attributes and that which have been identified by biologists. So we are back to our problem and we can move on to talk about our different kind of outfits for DNA. And the first outfit is no outfit, basically naked DNA. Now what do we mean by naked DNA? So here we've got a particular representation of DNA. Okay, so that's naked DNA here, so that's the double helix in plain water. And here plain water is not represented. This representation is experiment informed, but what we can try is to zoom in a little bit to get some molecular details. And so here we've got a bit more molecular details, and we see that there is this group with like one yellow bit surrounded by 3 red ones. It turns out that these groups, called phosphate groups, carry negative electric charge in water. And of course, if they carry negative electric charge, we need to understand what these actually do. So to do this, as a physicist, I simplify my problem and I say that this particular DNA double helix can be approximated as being a cylindrical core with a double helix of charges around it. Now, we need to have a step back to discuss what electric charges are and what they actually do. So it's simply a property of matter, like size or mass, okay? Now, the electric charge of a material object can be one of 3 kinds. So it's either negative, so that would be in blue here, or 0, that would be in green, or positive, and that would be in red. The 0 charge essentially, let's say, does not interact with the negative charges or the positive charges, at least not in any significant way, and therefore we discard it. 0 charge means 0 interaction. Okay? Now, the negative and positive charges, however, they attract each other. Okay? So opposite charges attract each other. So this is one of the rules of electricity. Now, when it comes to charges of the same sign, well, they tend to instead repel each other. Okay, so charges of the same sign repel each other. Now these two rules can be put together into let's say a graphical representation where we put either a negative charge or a positive charge in the middle of a field of arrows. And these arrows simply gives a direction of where a hypothetical positive charge would go if we were to put them in the neighborhood of these charges. So for example, if I put here a positive charge, then it would follow the arrows and be attracted toward the blue on the left hand side and repelled from the red on the right hand side. Now these arrows can be interpreted as being extracted or derived from a more fundamental quantity. And this more fundamental quantity is what is called the electric potential, which is basically some kind of landscape, like a mountain landscape, and the slope of which would be the arrows forcing positive charges to be attracted to negative one and to be repelled from positive ones. Now I want to get rid of these arrows. Some of you may remember things like this that I've done in the talk from last year. That's because we talk about charges again. Now, what I want to do is to get rid of the arrows and look at the three-dimensional landscape of this electric potential. The way I want to do it is basically by starting with neutral charges and then charging the left one negatively and then the red one positively. So you see, that's how it will progressively deform the actual electrical landscape to form a valley for the negative one and a hill for the positive one. Now the rule here is that positive charges go in the direction of valleys, so they try to go downhill. Let's have a look at DNA now. So let's assume we've got some neutral DNA in the first place, and it has some electrical potential which is entirely flat, so there's no landscape whatsoever. And then we again slowly but surely charge negatively the phosphate groups on this DNA. So how does that look? So that's how this will actually look. There are a few things we can notice here. First, notice that the height and color scales are the same as in the previous slides for the ions or for the charges. And so what that means is that bare DNA creates a very, very steep potential landscape. And in fact, DNA is one of the most highly charged, most strongly charged molecule in nature. So if we were to compare electric potential as if it was the same as light to our eyes, then for other molecules, DNA would be literally a molecular lighthouse towards which some molecules would run to and some others would basically escape from. Okay? So what would be the related functions of this naked DNA? Well, one possibility is to attract reading and compacting molecules from far away. Okay, so that's something that some, like myself and colleagues, have put forward about 7 years ago. It also affects the rigidity of DNA. So if you try to bend DNA, then you would have two neighboring negative charges that don't like to be close to each other. So they want to be basically bent back. Okay, so what that means is that it's difficult to bend DNA due to the existence of these charges. And finally, it also may enable target recognition. So when you read, then you need sometimes to get a very specific target, and then that's this is believed to be related to this very strong electric potential. Right, so that's for naked DNA. Let's have a look at the second one, which is camouflage gear. So where does this camouflage gear name come from? Well, so let's back to our DNA here. Okay, so this is our double helix DNA, or at least our toy model. And what we can, of course, notice and say is that DNA is not alone, especially not in the cell. There is, at the very least, water. Okay, so here these are water molecules. They would be here considered mostly neutral. And you've got counterions. And when physicists are confronted to a system where you've got all these charges here, these counterions, which are positive ions here, as they are red, you've got this DNA, and then you've got these water molecules. This is an incredibly complicated problem. They're hardly simulatable in a supercomputer. Like, it's really, really complicated. So what people do then is they do what is called coarse graining. Coarse graining consists in basically becoming, on purpose, blind to the water molecules. Okay, the water molecules are still there. They will affect, for example, the way the ions are moving around, but we, on purpose, become blind to the water molecules. But because they are affecting the motion of the ions, then we need to extract the new rules, some effective rules for the physics of the ions and how they move. So how does water affect ions? Well, there are many water molecules, many more than ions, and water molecules then will kick around the ions in multiple directions, all right? As a consequence, the motion of ions appears random. Okay, so here I'll just give a very quick illustration of how random that looks. And this gives indeed a trajectory which looks, well, everything but regular, right? It's entirely random. And if you were to look into it deeper, this is actually fractal even, etc. Right, so that's for how water affects ions, and then when we kind of forget about water. Now, how does DNA affect ions? Well, DNA is strongly attractive to positive ions, but we've seen before that the counterions I'm looking at are positively charged. Therefore, well, ions will be attracted towards DNA. That's one thing done. But how do ions affect DNA? Well, DNA is visible via the electric potential landscape it creates. But the thing is that counterions also create an electric potential landscape, and in fact they create hills instead of valleys. So as a consequence, ions will deform the potential created by DNA in the way that you can see on the image here. Now, if you put all of these things together, you will get this movie on the left-hand side. Okay, what we can see, or by summarizing this, is that ions tend to move randomly, but at the same time they are attracted to DNA, and they perturb DNA's electrostatic potential, as you can see here. As a result, we have that ions tend to stay very close to DNA, but yet they kind of move around, and DNA's resulting potential is substantially, really substantially less steep. In terms of physics vocabulary, we say that ions actually dress the DNA molecule, and the drop in intensity of the apparent potential of the dressed DNA is called electrostatic screening. It is believed, in fact, that up to 90% of DNA's charges can be screened in some circumstances, and then it would make essentially DNA invisible to other molecules. So what could be the related functions in biology that we're interested in? So it seemed that this could be related, so this very strong screening could be very efficient for DNA packaging in viruses or in our cells, of course, because you need to compact them, these things, to compact DNA into a very small volume. If they are too close to each other, they will repel. So of course, if you want to prevent this, you need to have this screening, okay? So that's one thing here. Another aspect that we proposed with some colleagues about 10 years ago was that this dressing of DNA by the ions could act as a protective layer, preventing a molecule to stick to DNA when they shouldn't stick to DNA, and they are looking for a target which is not yet actually found. And so in that case, this iron layer that is dressing DNA would act as a cushion, preventing these molecules to actually, these reading molecules to actually stick to DNA where they shouldn't. So the analogy would be to look at some kind of maglev train that would be going on a DNA track. And finally, some people have proposed that it would contribute positively to so-called homology recognition. So it has to do with repairing and gene shuffling mechanisms occurring in the cell very regularly. Right, so that's for the camouflage gear. Let's have a look at the last one. Okay, so the last one is the full protein armor, DNA. So why do we call it that way? So we call it that way because when we have discussed that this DNA was surrounded by water molecules and quaternions, that was the very minimum they could have. However, there are other things like huge proteins that can, of course, be around DNA. And this particular protein is super crucial for our DNA of humans. So it's called a histone core. A histone core. And this is something that is experiment based. And this huge monster, as you see, there are 8 different colors and they correspond to 8 different subunits. Now these histone proteins, that's what we're interested in, they are positively charged. So what does that do then? How do histone then affect DNA? Well, first, DNA's rigidity, we need to remember, comes from its strong electric charge. Now, if there are quaternions and histones that are positively charged and around kind of DNA, then they will actually decrease the DNA's attractiveness. So that's what we've seen before, like the landscape, but also its rigidity. And as a consequence, DNA ends up wrapping around the histone core to form a so-called nucleosome. So a nucleosome is an object made up of, if you will, histone core, so it's a protein core, and then that is wrapped around with DNA. So it has DNA and protein, and this thing is called a nucleosome. Now the story doesn't end here. In fact, many histone cores will bind with DNA to form a string of nucleosomes, and it is believed that then this nucleosome make up a complex hierarchical structure that efficiently pack DNA. This is what is called the chromatin fiber hierarchy hypothesis. And if you want to have a quick example, this is how it could look. So here you're going to see an artistic view or impression that is partly based on experiments about this packaging effect. So here you've got DNA, and there's been an histone protein that has, attacked DNA basically, and now they are self-assembling into a nucleosome. Okay, so that's what a nucleosome could look like. And then this nucleosome form a particular structure, okay, itself, which look like a thread itself. And this thread will basically form a 30 nanometer so-called chromatin fiber thread. The thread itself will form another additional structure, which itself will form a new thread of much larger diameter, as you can see here. And eventually it will give back, according to this scenario, it will give back the familiar, say, chromosome that we could see with a good microscope. So that's kind of the story, how the story could go. What could be the related functions? So I've got to go a little bit quickly here. So one of the functions, and that's why it's called armor here, is to protect against damage, DNA damage. So there are here, I put forward 2 papers, one that has to do with protecting against DNA radiation damage and iron-mediated damage, which is more like chemical damage. Another aspect has to do with acting as a librarian, and the idea is basically to switch off or on the access to genetic information. So, as a librarian, it's as if you could say, no, like somebody asks you a book and say, no, not today. Today the book is not available. So basically, the chromatin can act in this particular way. And then there is this idea that there are traits that can be inherited and that are not related to the actual genetic code, but which are related to like superstructures related to it. So like the how the nucleosomes are aligned, how packed this region of DNA is and not this one, etc. So this is a new trend in genetics, a very exciting one that is relatively recent, and it's definitely ongoing. Now I want to summarize the talk. We are reaching the end by again saying that we've presented these three outfits that basically satisfy or fulfill different roles for DNA. And I will try to summarize them with literally a couple of words. So naked DNA, if I have to summarize it, would be simply very attractive, right? Naked DNA is very attractive. But the camouflage gear enables DNA to become suggestively attractive. So it's attractive, yes, but only in some places, and where it doesn't want to be attractive, it's not attractive. Okay? And then the protein armor, I think a good way to summarize it would be to simply say it's protectively attractive. Okay, so there are some parts where it's fine to go to, but there are some parts that are protected by the chromatin and the nucleosomes, where molecules or chemicals can't even go. So that's it. And of course, I will have some closure comments. And this would be that, of course, it's tempting to be again amazed by how DNA is versatile and how it uses its environment very efficiently. But that would be really a one-sided view. We could also, this would be in particular overlooking the equally remarkable ability of ions and molecules to use perfectly and complement DNA's innate properties, and all of this to have basically functioning cells and functioning organisms. And on this, I thank you very much for your attention.